I just want to describe a little bit of what's the difference, because nobody knows the difference of one exosome versus another exosome versus another exosome. I always start off this presentation. I've been lecturing for about 15 years in the cell therapy sector. I'm from the cell therapy sector from University of California, Irvine. Over 24 years ago, I was part of clinical studies in spinal cord injury and stem cells. And I start off with this because it's ultimately the smallest units of life, and each cell in our body requires communication. Remember, every cell in your body, every single one of them communicates with another one to survive and to be in homeostasis all the time. These are those small little things called exosomes. And it's interesting because everybody says they have exosomes, we have exosomes. Yet there are a true definition of exosomes. And when you go talk to somebody, the first question, I'm a scientist, you should ask is, have you gone and looked at the standards that are out there, and have you identified the standards that show that you truly have an exosome? And these basically are a standard that is the International Society for Extracellular Vesicles, and you have to show two different markers, meaning you have to show that there's truly a nanoparticle there, which is a flawed system, and I'll talk to you about it in a little bit, and you have to show it by size, that basically it's in the right size, it's in 0 to 100 nanometers in size, and then you have to look at what it contains. So we look at on the outside of this microvesicle, we look at specific proteins, like the composition, and you need to demonstrate that your exosomes have at least 3 of these that are there, which is ALEX, which is TSG101, tumor suppressor gene 101. That's inside of a cell, excuse me, an exosome. And then outside of them, we look at markers like CD63, 81, and 93. But once again, not all exosomes are the same. One baby versus another baby versus another baby are completely different, right? And for those of you that have been talking to individuals that talk about I have a ground up piece of umbilical cord, there's stem cells in there, there's exosomes in there, those are basically individualized donations that were gathered from a baby. But guess what? Just like that baby developed, it's not going to be the same concentration. Think of this. Every single body in this room is completely unique. So the concentration of one product versus another product versus another product, when it's from, let's say, amniotic fluid, you'll see some out there that are amniotic fluid, you'll see it from ground cord tissue, it's not going to be the same. These are a lot of the products that are out there. You know, a lot of them are ground up tissue, amniotic fluid, not abundant in stem cells. They're just collecting these things and then putting them out there and selling them. The amount of testing that's actually done on this is very, very minimal, and there's variance from lot to lot. The lot productions are actually done in about 100 vials, and that's it. Anywhere from 100 to 150 vials, they're done. Depending on what it is. People talk about having exosomes from a plant. Nobody out here is a plant. Remember, we have completely different sequences than plants do, and even mice do. I've done a ton of mouse research, and guess how much mouse research actually translates into a human condition? Can anybody tell me the billions and billions of dollars that we do in research, and I've done a ton of it in spinal cord injury, in autoimmune diseases, in brain trauma, all of it. Less than 5% of everything that's done in the research in the lab ever translates into a human condition. Ever translates actually into a functional drug, which is even neater. So there's a ton of research, and we learn from it, but it doesn't necessarily mean that we are mice and rats. Think of this. People show you a study, this supplement works, it worked on this mouse, it did this, it did this. We are not mice, we're not rats. It does not work like that. Show me some human data, and that's what we're going to show you a little bit about here. So number one, an exosome is only as good as where it came from. Number two, the biggest thing is, when we produce these, for instance, ours are from an umbilical cord lining stem cell, and I'll talk to you a little bit about it in a second. When we produce these, we put them in a specific microenvironment. Everybody here is only as good as what? You're only as good as basically what you put in your mouth on a daily basis, your diet, your supplementation. It's the most important thing that you deal with on a daily basis. Similarly, when you grow up cells, it's the same exact concept. It's the same exact thing. Many of these, believe it or not, as you grow up these cells, they'll have genetic issues. You should be concerned about that because they pop up. We tested 50 different cords, we grew these up, and later on down the line, we would find genetic chromosomal aberrations. So this is an issue that you have to consider. Where am I getting something from? Is it possible that this is going to transfer to me? People still don't know that. So you have to be very, very cautious of this. Our product is derived from this. This is an umbilical cord lining stem cell. This is a publication of mine from 2010. I've been studying cells and exosomes since 2000. So the product that we have is published from here. It's a patented technology. These cells are actually in 4 clinical studies that are going into phase 2. Actually, one of them is going into phase 2, 3 at different universities throughout the United States. From there, we've derived, specifically tailored, a product, an exosome product for skin. When we look at why is one cell better than another, we look at things as, for instance, comparisons and how clonable are these stem cells. You look at something called colony-forming units, and you can actually see here on the very, very bottom the amount of dots. Those dots basically representing what you're done in a graph are basically how many colonies we can form from a single cell. That is the true definition of stemness, is one cell replicates into 2, into 4, into 8, etc., etc. The cells that we use actually in our clinical studies and for our product are what's called passage 4, and you can even see, even at passage 6 later on down the line, they mature just like we do. We culture expand, grow, pass, culture expand, grow, pass. Continue on. They age just like we do artificially. We use these passage 4 cells, which we have evidence that are actually 15 years of biological age. And how do we know? We're one of the only companies that do this. We test telomeres. Has everybody heard of telomeres here? Better have heard of telomeres, please. It's something that you can measure in your body. If you actually come close to being about 5,000 base pairs, you have critically short telomeres, you've got an issue going on, you better change your lifestyle to stop that trajectory of all the neurodegenerative diseases, cardiovascular disease, etc. So we look at this, and these P4 cells are about 8,000 base pairs. You were born with 10,000 base pairs. The neat thing about birth, from the time you were conceived, egg and sperm meet, you start off at 15,000 base pairs. In 9 months, we cleave 5,000 base pairs. The remainder of your lifespan, you only cleave about another 5,000. That's how many crazy changes transpire in this. That's how much development transpires in those first 5 months. So the cells that we have are about 15 years of biological age. We have evidence also that telomerase are actually activated. We've done, this is back from 2013 when we started looking at this product, how do we tailor-make a product that's for cosmetics? The first thing we look at is the second most abundant thing in your body, which is hyaluronic acid. And we looked at various different formulations and compared other stem cells here, bone marrow stem cells, versus ours. And we found a formulation that is high in high molecular weight HA. That's also a difference. Is the size of the molecular weight of basically HA is very, very important. You got to be conscious of this because this stuff breaks down. And when you're applying it on it topically, you want something that's going to be high molecular weight. So that's formulation after doing a ton of studies back in 2013. This is what our product contains. And I showed you the definition a little bit ago of an exosome. I'm showing you right here that CD9 is present over 1,500 nanograms, CD63 is present 128 nanograms, 81, and then the intra-exosome, TSG, is that. But the most important aspect of our product is we look at what we did to the cells to make sure that they are secreting out the most abundant protein in your body, collagen 1 and 3. It's there, naturally occurring from a stem cell. Hyaluronic acid in high molecular weight, it's there. LL37, does everybody here know what LL37 is? It's a peptide and it's naturally found in your body. It's an antimicrobial, one of the strongest antimicrobials. When used correctly in nanogram amounts, there's a significant amount of evidence that it helps. But in the context of skin, those of you guys are all experts in skincare. What transpires with skin a lot of times is you get a lot of microbes, right? So what does that help with? It helps clear the microbes, and it helps clearing of skin, clearing of blemishes, clearing of acne, these type of things. That is one of the products that's actually naturally occurring in there. And then we have some anti-inflammatories and some other mitogens that are actually out there, growth factors. When we talk about growth factors in this product, you talk about other companies talking about things, and scientists know this, that an exosome can be stable at room temperature, not very long. Because just like these growth factors are in your body, we're running around at 98 degrees, 98.7. Remember this, when you're room temperature and you're fluctuating up and down, and we know this at lab, we keep growth factors frozen until they're ready to use, because guess what? They break down and your cell produces a new growth factor. On a consistent basis, this is transpiring daily in your body. So when people talk to you about an exosome at room temperature, that's survival, that's nonsense. And I have no problems with refuting this with another scientist. It's complete nonsense. We do this all the time. We do culture all the time. I've been culturing cells for over 24 years now, and you can see this actually. Our product contains a significant amount of amino acids, and the concept behind all these amino acids and all these proteins in there were actually to make it stable. We've measured this, we've looked at this. Our product's completely different, specifically designed for skincare more than anything else. And then once again, I'm showing you one other aspect of, I'm showing you through flow cytometry of, we have exosomes. This CD63, CD9, this is done through flow cytometry. You want to ask the questions, what is the science proving that you have an exosome? And this right here, somebody will tell you, you guys have walked around and talked to people about exosomes, I have 1 trillion exosomes, I have this exosomes. This is from a machine called a nanocyte. And the nanocyte reads particles. And when you look at the way this reads particle, we actually measure them, look at the particles, and we get a read on the particles. But when we get this read on the particle, being a true scientist, you question, at first when I got a read, I questioned we have 300 billion exosomes per milliliter, and my questioning was correct. Because then I send controls, which consisted of saline solution, which consisted of Hank's basic salt solution, and guess what? Those simple solutions measured about 1 billion particles per ml. These are just a carrier. So depending on what you put it in is what's going to register and backfeed. A simple example is such as amniotic fluid. Amniotic fluid is basically consisted of 55% antibodies in there. Antibodies fall into somewhat of that spectrum of where you read nanoparticles, and they can say it has 1 trillion particles, but it's not true. You normally want to bind something on the outside of that double membrane of the exosome, and then you want to quantify that, which is what's done here. So look at the big difference of, we have 50 billion particles right here with the red bar, versus if you look at the other one, we have, it's about 200 billion particles. It's a big difference, fourfold difference. And that's when you run a standard one without running the fluorescent dye. And even with the fluorescent dye, the machines are not accurate. We're getting inconsistent results. Our results we looked at before we put this product out, we actually did skin studies. We looked at skin, and most importantly, what we wanted to look at is, what does the product do? When you use it in small amounts, you can actually stimulate your own collagen production. You can stimulate your own elastin production. There's this data here. You're welcome to stop by our booth. We can give you physical copies of this. You can actually take a look at it and register. And I'm always there to help to look at, ask any questions or anything. The other thing is that there's questioning on taking a product from a liquid form and actually putting it in lyophilized. So freeze drying it doesn't really work, and do exosomes survive? Yes, they can when done correctly, which is something that we've done. We spent 2.5 years finding a formulation that'll stabilize it in a powdered form. However, people still prefer putting this and using it in a liquid form. Because in liquid form it comes frozen, you just transfer it, you don't have to dilute anything, you're just ready to go, and you can use it that way. But depending on what you want to do with it, this is another method that you can actually take it. For instance, here's more data on looking at the data of how we came up with our formulation of our lyophilized product that you can actually see clearly. The most important aspect that you guys are seeing in here, what is everybody consistently talking about? Collagen stimulation. Remember, it's the most abundant protein in your body. We're talking about as you age, this stuff decreases significantly and it breaks down. I'm presenting you with a product that has it naturally and helps stimulate your own collagen. There's so many different types of collagen. HA, everybody talks about, I want to use HA, I'm going to glide HA, I'm going to use different forms of HA to apply it on. I'm talking to you about a product besides having exosomes in it, has naturally occurring HA, which is key, and it's high molecular weight HA because we've actually tested this. Because here, not like the penis, size does matter. So this type of exosome is really, it's on another level. You're going to see a bunch of competitors here. You'll talk to a lot of other people here. We've been doing this for a really long time. Safety, we focus on safety. We have a lab. I'm actually out in Corona, California. For those that are in SoCal or your business SoCal, please look me up. please come over to the lab. You're welcome to come in. We do a bunch of different cell therapies, a lot of different work that we actually do there, a big group of scientists. You're welcome to come check out the lab. We do an extensive amount of testing. The 3 cores that we have to produce this product are from mothers that are completely viral free, which is another interesting fact, because 85% of the patient population walks around, 85% of us that are in here have either CMV or Epstein-Barr virus. Epstein-Barr virus, for those who don't know, is one of the worst viruses you can have. It's mononucleosis, the kissing virus. But some people can't sustain this. It causes cancer, it causes autoimmune diseases in the long run because the immune system becomes challenged and tapped. So right now, people will tell you, you'll go to a booth and say, my product's approved. I got to call Bing BS. Okay, sorry. There is no approved exosome products out there. The FDA, you can look it up clearly, go onto the FDA's website. There is no approval. Current good manufacturing, when people say they practice it, if you have the correct system in place, you practice a regulatory system and you build a current good manufacturing practice, because I know this because we have clinical studies in place, and for you to get certified, an FDA has to come and visit and give you that certification. Not an outside entity or anything like that. People talk about I submitted a master file to the FDA. It doesn't matter. It's a document saying this is what this is made of. You need an investigational new drug application to show that they approved you moving forward in a clinical study. Even with that, people say I have an investigational new drug application in a product, I can use it and sell it. Doesn't mean you can sell it. Think of the simple term, I synthesize a foreign version of cocaine and I run it through a clinical study and I decide to snort it and sell it on the side. It's not approved. It's not approved. It's not approved until you show it does what it has to do. Protein is key to function. People talk about messages, etc. What makes changes in our body is protein. The cell takes a message in, it synthesizes a functional protein, and it's what makes a change within our skin, within everything that we have. Just to show you real quick some results. At 2 p.m. we're going to have some live demos over there. I don't know if they can pick up more people to actually do live demos. But Dr. Kwok will be doing some live demos right next to the roll over from here. We'll have some live demos with the product. You can actually take a look at the way it's actually used. Following microneedling, we're going to be using the Candela device. Here you see some more, basically before and afters. And this is exactly how we use the product. We have these sprayers at our booth because this is an atomizer that uses a lot less product. It atomizes cleanly, you spray it on, and the product absorbs relatively immediately. It's a great way to apply it and use a lot less, where you can run many throughs. So the practice that we do is you spray, you wait probably 20 seconds, it dries, and then you do your procedure and you spray again, and you may do another run of procedure, which is very typical, and then you would spray again. With 1 milliliter of product, you can cover an entire face easily. So the difference with us is superior exosome with strong scientific backing. We've done this for many, many years. It's made in the United States, not made anywhere else. Product's made in Corona, California. Come down to our lab, like I say, with no problems. Contains healing properties because we've done it that way. We've studied it for many years. The difference with us is we are a company that's been in existence for 16 years. Before we got here, we spent $26 million to get on the research portion of it alone. So it's a little bit different. Uniform dosing because we make lots that are consistent. The protocol is run on a consistent basis, and we produce lots of vials that run about 5,000 to 6,000 vials for one lot alone when we do these, a specific filtration system. Ours are from pure mesenchymal stem cell, or what we call umbilical cord lining stem cell, and our cells are part of 4 FDA-approved clinical studies, of which 1 are going into phase 2-3 study. Come join us if you want to see for yourself how this works. From 2 to 4, we'll be doing some demos right there. I appreciate everybody's time, and it's an honor to talk to you. You can reach out to me at Dr. Gonzalez at Exo. I'm always open for questions, help, anything I can do. I do cell therapy, and it's what I love to do. You look me up, you'll see all the different cell therapies I work with. I'm here to help. Thank you very much, everybody. Pleasure.